ecological succession was. But we've also got a few main types of succession. Can anyone tell me what those are? Any ideas? There's two that we will be discussing in class, and then the third we'll be doing the lab. But we've got primary and secondary succession. So can anyone tell me what primary succession is? Any ideas? Pardon?
When there's no soil.
It's when there's no soil. So yeah, it's when plants are growing in an area where no life had been previously. So what I've actually done here is on Webspiration, I just prepared a quick little storyboard. This is something that we could do, I think, with our students. We could compare primary and secondary succession. It works quite well. You can throw in these pictures and just throw in little blurbs along the way. Yeah?
There's quite a difference between where there's no soil and where there's been no life previously.
Right, so it's like on rock is a good example. So after, say, like a volcanic eruption or after a glacier is receded in an area such as that.
Bare earth.
So yeah, so bare earth. So if we follow along in this storyline here, we have a volcano somewhere in New Zealand's tropical Northland erupts. As the lava cools, the lichen will grow on this newly exposed rock surface. And then as this lichen dies, it starts to form a bit of humus. This humus will allow some small plants to take hold, like mosses and grasses, eventually ferns. And these will grow down further and break up the rock. And I'll just get rid of this here. Got to update my virus protection. So once a little soil has been made, pioneer tree and shrub species like Manuka will be able to grow. These shrubs and small trees will provide shade and more soil and allow other shade-tolerant species to grow up, like the kauri or the reemu trees. So that's just an example of the storyboard. I was thinking it would work well for this kind of thing. And if you look on your handouts, after we would go through primary and secondary succession, we could fill out this Venn diagram as well. The students could work in groups, discuss the differences, the similarities, and then fill out that diagram. And so for the purpose of today, we're going to talk about small-scale succession or decomposition. So this is when organisms will feed off a dead body, rotting log, or food, even feces, and different organisms will move through in succession. They're basically preparing the environment for another species to move through. So if we look on this last slide here, we've got a picture of bread. This bread's got a whole bunch of different mold growing on it, and this is kind of what we're going to get into today. So if you turn to the on the back of your first sheet there, we've got our aims and our procedure for this experiment. So what I'm going to get you guys to do is we can break up into, judging by what we have here, maybe groups of 4, and we're just going to prepare a sample. So this is how we would prepare the experiment in class. And then I've also got some examples here that have been growing mold for 3 weeks, 2 weeks, and 1 week, and I'll just get you guys to cycle through those as we prepare these samples. So if you follow along on the method on your sheet, we're just basically going to be cutting the bread into squares large enough to fit into this petri dish. And because this loaf isn't pre-sliced, it's also got to be thin enough to make sure that we can close the petri dish. We're going to have one sample that's moist. We don't want it to be soaked, but we want it to be moist. And the other is going to be left exposed, and it's going to be dry. And we're going to be comparing the rates of succession for moist versus dry bread. So if you guys want to break up into groups now, we'll start to get that ready. And in terms of safety, guys, while you're preparing the bread, just be careful with the knife. Keep your fingers away from the blade. And yeah, once I show you guys the samples, you don't need to take off the lid. You don't want to be breathing in the spores that these lids are giving off. So just keep that in mind. Yeah, I'm going to actually put one on each table, and then we can cycle through it.
Interesting colonies growing.
out your samples, you can come around and have a look at these samples that I've laid out here. And you can give a go at drawing out your diagram and maybe trying to assess the percentage cover of the different molds. Sorry, I'm just going to get you guys' attention for 1 second. Everyone? Yeah, thank you. So we're going to, I've laid a sample on each one of your tables. So once you've prepared your own, you've got your white bread and your dry bread. You can have a look at the one on your table. You can cycle around. I think you guys have one that's 2 weeks old. At this table, it's 3 weeks old. And at the back table there, it's 1 week old. So you can, we've got markers there. You can do a diagram on the data sheet provided and try to assess the percentage cover of the different species that are on there. I have briefly done a bit of research on the different kinds, so I'll help you along a bit, but I'm not a mold expert. So there should be, I have a container here of water. Yeah, sorry guys, I forgot to mention. I have a container of water here with all the other materials, and there's little plastic pipettes here as well that you can drop your water on your bread with. So just enough to make it moist. Yeah. And then, so the idea is every couple days we'd be checking these bread samples. We'd want to be taking notes on your data sheets. You have a spot for date observed, and then the notes and a place to make your diagram. So the idea would be every couple days we'd be tracking the progress of the growth. Yeah, so the dry one is exposed and the moist one will be open. Yeah.
So that's 3 weeks of dry.
That's 3 weeks of dry, yeah, nothing's happened. It's shrunk.
Like a cracker.
Yeah. So that's the exposed one. So they've been exposed, they've been prepared at the exact same time. But this one's moist and this one's dry.
What would you say, 100% correct?
Yeah, I think so. Right. Yeah, this is fluid. But I guess the idea is to keep it moist and has to be stuff.
I wonder if
And so, like when these stores